what prime editing is and how is it differentiated from nucleus editing and from base editing?
Sure, I'd be happy to. Of course, that would take the rest of the 35 minutes that we actually have in order to do it justice. But prime editing is really what we would call a third-generation gene editing approach after nuclease editing and base editing. In each case, each of those techniques is sort of improved upon and solved some of the problems that were there with earlier therapies. Prime editing has really been a giant step forward because prime editing really has four characteristics we like to highlight. It's versatile, it's precise, it's broad, and it's very efficient overall. And what do I mean by each of those things? So versatility is really prime editing can correct as well as to destroy genes. So CRISPR-Cas9 generally is most effective when it really removes a toxic insult to the cell by destroying it. Base editing begins the process of correcting by changing some of those base pairs that might be wrong in a mutation, but it can only do a limited number of them. And both those techniques are extremely good as long as they stay within their area of focus. Prime editing just goes well beyond that. We can fix all 12 types of base pair mismatches. We can correct small deletional mutations, insertional mutations. We can work with hotspots, areas of 100 or 200 base pairs where mutations cluster. And in the last year or two, we've been able to very precisely remove, any DNA that we would like to remove. A good example is removing the pathological repeats of triplet diseases or repeat expansion diseases. But in the last year, we've also highlighted some of the work where we can put in kilobases, thousands, tens of thousands of base pairs, very precisely into a particular location in the genome as well, which really differentiates it from other ways of getting DNA into the genome as well. So that's the versatility. It's the breadth of what we can do. The second thing is it's extremely precise. When we go in, we make an edit or we leave the DNA unchanged. And that's important at the site that you're editing, where you get very pure edits or you get nothing. But it's also important for off-target editing, where everything that we've done so far suggests that prime editing almost certainly has extremely low levels of off-target activities. Certainly in head-to-head comparisons, it appears better than with CRISPR-Cas9, and there are many reasons why it may even be superior to base editing. It's a beautiful technique because it works at the site of the actual gene. So we make our corrections, we leave things under endogenous control, but permanent corrections, all the daughter cells get them. And it's extremely broad. I explained the types of things we can do, but if you actually look at rare genetic diseases and you ask, hypothetically, how many can we correct? We can correct well over 90% of known pathological mutations. Now, of course, that's only in the sphere of rare genetic diseases. Prime has many things that we're looking at. They go well beyond just those rare genetic diseases.
Got it. Thank you. That's a very helpful overview. I want to hone in a little bit on the better specificity of prime editors. I think you talked about three edits or three edit checks in the past. I would just want to make sure those edit checks doesn't have completely the same sequence, so that it's not independent checks anymore. How different are those three checks?
So the three edit checks are different. So there are three independent edit checks. Let me just explain what that means. One of the concerns when one has off-target editing, which is, I think, a major concern for any kind of gene editing type of approach, just to set the stage, you want to be able to say to the patients, to the doctors, to the regulators, you go in, you make that therapeutic edit, you get out, you don't muck around anywhere else when things happen. And of course, with all of these editors, particularly the ones that are CRISPR-Cas9 or other types of CRISPR approaches, you always have the risk that you may land in the wrong spot and that you may do something when you land there. Prime editing is different than many of the other approaches. We don't cause double-stranded breaks, okay? Which are emergencies to the cell and often result in a lot of mucking around with the DNA. We really just nick, and we share that with base editing. But one thing that's a little different, I think, that you referred to is it's a three-stage process. And at every stage of that process, there has to be an annealing or a matching step that occurs. So I really use the analogies of it's a door that has three locks on it, and if you don't unlock each of them with a key, you really can't get to the other end. And so prime editing has a lot of specificity. And you really ask the question of, are they really three independent locks? And the answer is, if you dig down in the details, there is one of the two of the locks that have some overlap, but also have unique elements as well. So there really are three independent annealing steps that have to occur because you have to have a really high degree of match across the whole annealing process to move to the next stage. And so in that three-stage process, you really can be sure if you land at the wrong spot, the chance that all three of those are going to be completely wrong is very small. Now, of course, one might worry, you're very precise, but are you actually efficient? And in practice, when I joined prime editing, it was one of the concerns I had is how efficient could this process be? But now we've really shown in program after program, we can get 60, 70, 80, 90+% precise editing in our clinical programs. And, it is turning out to be an incredibly efficient approach as well.
That's very good to hear. Not just specific, but also efficient, and that's not an easy task.
No, it
to
isn't.
achieve.
And frankly, part of the reason is that prime editing today is greatly different than it was even 3 years ago. There's just been tremendous improvements. Many have come out of the Liu Lab. Some have been in the literature because prime editing is something that's regularly published on by David Liu, one of our founders, by members of Prime, also by a very large community. So everybody gets to see how to improve prime editing. Many of those we've taken into Prime. Some of them are proprietary. Some of them are more open. But they've really improved prime editing across the process. And that's really changed it from what was an incredibly powerful scientific technique to one that's really a therapeutic technique that has the high level of editing efficiency and the extremely low level of off-target editing that we're really seeing in our programs that are moving towards the clinic.
Speaking of continued improvement, I think David Liu last week just published a new version, PE6. Is that something that you have access to?
Yes, it is. Pretty much everything that David's done has come through directly to our laboratory. It's very interesting. If you read the authors on many of David's papers over the last couple of years, they include many folks who have joined Prime Editing. So David's been kind enough to point, I think, 7 or 8 of alumni from his laboratory, some of the brightest postdocs, towards Prime Editing. We've been lucky to get them. Though, of course, we've got many people from other locations as well. But they've come in, so they've been the co-authors on many of that work that they started in his laboratory. And when they came into the laboratory, they were already expert in that approach. But of course, we've managed to bring a very, very strong IP position in all of those improvements as well, really to wall up Prime Editing as something that really belongs and is really proprietary to Prime Medicine as well. We think that's very important in addition.
Okay. Maybe a couple of technical details. The length of insertion and the length of deletion you can do with Prime Editing. Could you give us some sense of that? And also, how big is the Prime Editor? Is there any issue with delivering this Prime Editor to cells?
No, they're both great questions. We get asked them quite frequently, and I love to explain the answer. Today, we can precisely remove literally 10,000 or more base pairs, and that's just the limit of what we've actually tested. A good example of that is some of our repeat expansion disease, our triplet diseases, where people may have thousands and thousands of triplets that really cause the pathology of those diseases. Diseases like Huntington's disease, but that really rarely has thousands of repeats. Some of the other repeat expansion diseases do. And we're able to take them out very, very precisely in that process. And we've done it in other diseases as well. Putting things in, we have mostly worked in kilobase-sized pieces. The example we gave recently at ASGCT conference was putting in a 3.5 kilobase piece of DNA. Again, very precisely. So when we put it in, we pick to the base pair where it goes, put it in there in the correct orientation as well. But in some of the papers that were really described showing, sort of demonstrating the technique, David Liu, for example, showed a 40 kilobase insertion in one of his papers that really described that overall. So it really gives you some idea of just how powerful this is. David loves to name and number things. So we now have PE1. We don't use it anymore. PE2, PE3, PE3B. Out of Prime Medicine, we haven't spoken much about it, but we have a PE3C. Then we move to Max, and the numbers just keep changing. The most recent work was PE6, but actually PE6 in David's nomenclature has 6 or 7 flavors. So there's an A, B, C, D, E, F. It gives you some idea of the complexity. Think of each of these as a tool to approach different kinds of genetic possibilities. And what we're building is a huge toolbox of ways that you can approach different types of problems that you're trying to solve. And essentially, those toolboxes are things that we use regularly across our programs. Now, PE6 is something that we clearly have heard about and been working on for a period of time. And once again, some of the authors on those papers are people who are now at Prime as well.
And in terms of the size of the editor, would it be any issue?
So the size of the machinery, to all intents and purposes, is very similar to CRISPR-Cas9 and base editing. It's just a little bit bigger. The thing that's probably the most important to highlight is the guide RNAs, which are the piece of RNA that both help find the spot and provide the template for the correction. Those can be larger. So typically for base editing or prime editing, those are about 100 base pairs. Ours are probably a little closer in traditional prime editing, 150. Of course, the greater you make those guide RNAs, actually the more template that you can add to really improve the ability to reach different types of corrections. And a major focus of ours is to be able to make bigger and longer guide RNAs because it really improves our ability to do different kinds of edits. Even so, when you think about the type of machinery they do, that extra 50 or 100 base pairs is just a drop in the bucket compared to the length of the machinery, which are used by all of the CRISPR programs and base editing programs.
Got it. And then lastly, on the platform, I wanted to ask about ability to detect off-target, because you don't make double-strand breaks. Of course, that's a good thing. But for someone asked to do the task of identifying off-target, this is actually a difficult proposition if you don't make any double-strand break. How are you identifying your off-target?
Yeah, so it's a great question, and in some ways, it's a great problem to have. In certainly the work we've done, which I want to stress is very preliminary, and it's going to take work like this for many years for me to be able to say with great assurance what I'm about to say. So as long as it's taken as this is where we stand today, our preliminary data suggests that the amount of off-target we have is literally minimal. As a matter of fact, in our first IND program, so far we haven't identified with the limits of detections of our assays any off-target activity. We're going to be doing a lot more work to help support the IND. It's not going to be a surprise if we find an example of it, but today we have none. That's a wonderful problem to have, as I said, but it does have a little bit of a problem. We expect that the FDA someday will say to us, in humans, when you do your editing, is there a spot you can look at that you've identified in your preclinical studies where off-target editing occurs at some small level, okay, some small degree? It may not be impactful, but it's a spot that's particularly prone with a particular prime editing. And can we follow in the clinic? Can we take a blood sample every X number of months, 6 months, and just make sure that in the folks that you're actually editing, that no evidence of off-target activity occurs in humans? Exactly how they may ask it, I really can't comment on today. But if you don't actually have such a spot, it's very, very hard to actually follow it. And we do know that some of the other gene editing companies have been asked to follow specific spots where off-target editing has been seen. We have some thoughts about how to solve that problem, but as I mentioned, it's generally a very good problem to have that we can't even point to an example of where we've actually crossed an off-target activity that we can be sure, or we can be assured of with any degree of certainty.
And when you do your off-target characterization, of course, one of the steps is to see how many places the genome has been touched. And with double-strand break, you can capture the break and easily identify here, and there are touched. And of course, that's in a model cell type and what, et cetera, and actual editing at actual dose level may not replicate that. But that gives you a framework to know the possible places that your editing can impact. Without a double-stranded break, how did you go about and nominate those potential places?
So first of all, we do the double-strand break assays because while we don't believe that prime editing causes double strands, I think previous early papers show that if you aren't incredibly careful, occasionally a double-strand break can occur. So really, that's something we work very hard to optimize out, but I don't want to suggest that if you were a laboratory today and were doing your first experiment of prime editing, you wouldn't see a touch of double-strand breaks. It's a very important part of the optimization process. But you're absolutely correct. We do those assays, and we expect to find essentially nothing to the limits of detection that we can do, and that's what it appears to be. But we also are developing what are called nicking assays, and nicking assays, because our prime editors do nick one strand of the DNA, which also identify places where things can happen. Those assays are more difficult because nicking happens in a more physiological way. So no matter how you do the experiment, there are always going to be some nicks there, even in your controls. But at the same time, what we can do is we can go in and show that those nicks don't happen and reoccur over and over again at the same location, which would build a case that maybe it was the prime editor that was involved with it, and it would give us a spot to look. But once again, the nicking assays so far have been very reassuring.
Got it. Let's pivot to the pipeline program.
Sure.
For the CGD program, I was just wondering, this is the ex vivo program, wondering about the minimum level of editing that could be expected to have a therapeutic effect there.
Yeah, no, it's a great question. For every one of our programs, as you can imagine, we do put a target on the amount of editing we expect to see in vivo to produce a therapeutic effect. In many cases, we cannot speak with absolute certainty. The data just doesn't exist because no one's really done anything like we've done before in many cases. But we can make some pretty good arguments about what that level should be. CGD in some ways is an exception to that because in this particular case, CGD is a very bad disease of childhood. People can't fight off infections, and they literally spend their whole life infected and often die quite early of those infections. In that particular disease, the only curative treatment is an allogeneic bone marrow transplant. So someone else's mismatched bone marrow into you to do the correction. And allogeneic bone marrow transplants don't necessarily take over completely for the bone marrow that someone may have. So there are a number of cases where people have had chimeric bone marrow, some of the old mutated bone marrow cells, and some of the new, hopefully curative bone marrow cells. And in those cases, if you have 10% or so, maybe up to 15% of the new cells populating your red, white blood cells, it seems to have a very important therapeutic effect. So we're arguing that 15% correction would be more than enough to get the therapeutic effect. Of course, we greatly exceed it. In those studies we've been doing preclinically, we're hitting 90 to 95% precise correction overall. And we've done that with donor after donor after donor in the ex vivo work. So we're greatly exceeding.
Got it. Sounds like that's a great first indication. Very low bar, although it doesn't sound like you need that low bar. You could achieve very high editing efficiency there. Are there more prevalent hematologic conditions that can be addressed with prime editing, but not with nuclease or base editing?
So the answer to that is yes as well. So first of all, it's important to emphasize that anything that you can see a nuclease and a base editing procedure to do, we can also do with prime editing. Essentially, if you think of it as a Venn diagram, we encompass all the things that they can do. Our general goal hasn't been to go head to head with those companies, but to focus on places where we can do things that we think no one else can do primarily. But yes, there are. The problem with ex vivo editing of stem cells is today, in order to get those stem cells back and to take over the bone marrow in humans, you have to use a fairly myelosuppressive regimen. It's mostly busulfan that's used today, but it's a very toxic bone marrow procedure. People are at risk for long periods of time. Many people feel, and I tend to agree with them, it's a real barrier to being able to convince people to get a bone marrow transplant unless there's no choice. CGD is a perfect place because the only choice you have is a worse bone marrow, an allogeneic one instead of an autologous therapeutic one. So it's a great place to start with this. We think we're on the path to solving that problem with an approach called shielding. It isn't an approach that's unique to prime editing, but we think we've joined with the premier shielding company, which is a company called Simeo, and we announced the deal recently. And what shielding does is it basically says, let's develop antibodies against all stem cells, human stem cells or hematopoietic stem cells, and then let's figure out, can we actually make an edit that protects the cells from that antibody's action? So in that process, what we do is we make a prime edit, and those new cells, once edited, are shielded from the action of this particular antibody. And so it's a process you can do a much gentler approach in terms of clearing out the bad cells that you want to remove, because it's very specific, and you never touch the new cells you put in. And since we can multiplex, which is something that's common not just to us, but to other gene editing approaches, but there's some nuances of things we can do that we don't think others can do. But since you can multiplex, you can do the shielding edit and the therapeutic edit at the same time. That work that work together as a partnership. We have to do some more work on it to get ready for what I jokingly call prime time. But if we're able to do that over the next many months or year, I think that will open up lots of other places. And we have a long list of other hematologic approaches that we would like to think about doing if and when it turns out that the shielding approach means that the bone marrow transplants are going to be much less of a barrier to moving forward.
Right. So the shielding is a milder conditioning, essentially.
Correct. In fact, hypothetically, it could be almost as if there's no conditioning, because you can just take out the bad cells. You could theoretically just put in your new cells at the same time, okay? And people, literally, the two will swap places. I'm sure it'll never be quite that simple, but hypothetically, to all intents and purposes, you really wouldn't need to do conditioning in the sense we talk about it today.
Great. Thank you. Let's switch gears yet another time to repeat expansion diseases. And this is to me, it's a very exciting area, very difficult diseases to treat. And yet you have several candidates in the pipeline, right? So these repeat expansions can be very heterogeneous between patients or even between cells in the same patient. Could be 1,000 repeats, could be 200 in the next cell. How do you achieve consistent editing outcome in these heterogeneous situations?
Yeah, so the repeat expansion diseases, we believe, are just an extraordinary opportunity for prime editing, perfectly suited for them. First of all, of course, our goal is to do what I'd call a genetic cure. I use the cure very specifically in the sense of basically bringing the DNA back to wild type or normal. Only with clinical studies can you show it's actually a cure for a disease. But the idea would really be that we could do a genetic cure for these diseases. And the reason why prime editing is so efficient in that is one of the techniques you can use, it's not the only technique, but one of the techniques you can use, something called double flap editing or twin editing, allows you to literally put your prime editors on different strands, bracketing the repeats. So in that particular approach, what you typically would do is you would remove, by being outside the repeats, all of the repeats. And then you can put back whatever you like in its place. So that means that whether a cell has 200 or 2,000 repeats doesn't matter, because the flanking regions are identical in each of those cases. And what you've done is you removed everything in between. And we've learned that it's just as efficient to remove 200 repeats as it is to remove 2,000 repeats, even though that seems a little counterintuitive, probably because the repeats form a secondary structure. And in fact, those two flanking pieces are actually quite close to each other, regardless of whether it's 200 or 2,000 repeats. So we loop them out, and we basically do it's almost like a surgical cure, literally as if we took a scalpel as the image I use and cut out just the repeats we don't want. What you put back in is really up to the science. In some cases, we do what's called a clean repeat. We basically take out all the repeats and we just leave repeatless. And we do that because in a few of these diseases, there have been cases where repeats continue to multiply over the course of an individual patient. And so if you take out all the repeats, there's no ability for them to keep coming back and expanding again. There are other diseases that it seems as if it's much more stable, and so we would put back just the missing repeats. And a good example of where that's important is something like Huntington's disease. I think you were going to ask me probably a question about this, Yanan, but in Huntington's disease, it's part of the coding region. So people really wouldn't want, if you do a genetic cure, you want to leave just the physiological number of repeats. If you and I have 5 of them, that's what you want to leave when you're done. And so we design it in exactly that way to leave just those physiological repeats. Now, of course, when you're dealing with repeats in the intergenic regions, in the 5' or 3' untranslated regions, you don't have to worry about getting everything perfect because those are untranslated regions. You just have to get rid of the repeats. Obviously, with cases like Huntington's diseases, and there are others where they're in the exons, the coding regions of the gene, you have to get it perfect. And that's something where prime editing is very good as well, because as I mentioned to you, we go in, we're incredibly precise, and so we either correct it to the base pair or we leave the DNA alone. And of course, that just means we have to get to a therapeutic number of cells where we've done the correction and we really can stop the disease in its tracks, or at least that's our belief.
Got it. And the first program is the Friedreich's ataxia. I was wondering why it is chosen as the first program. As you mentioned, Huntington's, there are other diseases that are probably more prevalent. And also, maybe please talk about delivery. For these different diseases, maybe where you need to get to is different. And are you comfortable with your ability to tackle these different deliveries?
Yeah, so we did not pick Friedreich's ataxia as an example to go first. We literally started with 7 of these repeat expansion diseases. Many of them are moving forward. That one just moved fastest. And some of it was there are beautiful assays to really show what you need to do. The science is particularly clear for Friedreich's ataxia. So that I wouldn't say we picked it as our lead as much as it picked itself to move forward. But there are others, myotonic dystrophy, Huntington's, ALS, that are moving forward incredibly rapidly as well. The second question, I think, was our ability to deliver it.
Right, and to each of the indications, it may be different places in the brain.
Correct.
Right?
So some of them are different places in the brain. In some ways, Friedreich's ataxia was important because there are some important tissues that we know we can get to today. For example, the DRG, the dorsal root ganglia of the spinal cord, are major foci of that particular disease. And we already have drugs out there that really work through that particular organism, that can get to that organ. SMA is a good example of that. So it was high on our list, knowing that we could find ways with certain kinds of AAVs to get there. Friedreich's is a more complicated disease. Just getting to the DRG doesn't cure all aspects of the disease, so I don't want to suggest that's the solution. Some of them are in muscle. Some of them are in more localized spaces. One of the repeat expansion diseases is in the cornea, for example. Some of them we think we're okay. Local delivery to the cornea we think is going to be relatively straightforward. Not easy, but straightforward. Getting to all the places in the brain, it's very clear there's much more work that needs to be done. I'm not satisfied with our delivery approaches, but part of what we're doing with those diseases, we didn't really expect that all of them were going to be in the clinic next year, and we haven't, of course, promised that. But we expect that over the next couple of years, there are going to be solutions, either from ourselves working on those diseases, and we have a lot of efforts there, or alternatively pairing with companies that have excellent delivery approaches as well. And we've been talking to a lot of those companies, and some of them have very exciting approaches. But I think the delivery to the brain and muscle is going to continue to be an evolving space for the next year or two.
Great. Perhaps we can touch on PASSIGE. This is, I think, an approach. You insert a landing pad and then insert your gene of interest in there. And this is used in CAR T, but CAR T is not listed in the pipeline. Is that mainly a partnering opportunity? And what is the advantage of PASSIGE compared with other ways? There are other ways to insert, even with targeted insertion into any location.
Well, I'm not 100% certain. I really agree that there's great ways to target DNA today into the genome. Most people who target DNA into the genome today use lentivirus, which is semi-random in its approach. I wouldn't say that's always true, but I think that there are some very significant problems with getting very precise locations of DNA today. And I think this is an example passage where you can show the power of prime editing outside of just curing rare genetic diseases. And I mean that two ways. It opens up a new field like CAR T, but it also shows prime editing can be combined with other techniques to really be synergistic in that regard. So as you pointed out, what PASSIGE does is it puts a landing pad for a recombinase in a particular spot in the genome. We can do that very, very precisely with extraordinarily high fidelity. And then by putting in a DNA donor with the correct recombinase site, we can literally put any piece of DNA, large or small, into that particular spot. That was described to some degree in the original paper. We've been working on it for years and years, but mostly under the radar overall. But I think it's that specificity of what we do, but it's also the fact that it can be delivered in one-stage, non-viral approaches. So we can, for example, edit T cells. We hope soon we'll be able to edit NK cells, and we can make extraordinary amount of edits in a single step, not using viruses, with very robust cells that come out of the process overall. The first place we've shown it is to make some CAR-Ts that we think have some significant editing advantages in terms of autologous and allogeneic CAR-T moving forward, and we're pretty excited about that. By the way, it's only one of a number of under-the-radar programs that we currently have that we've spent less time on than some of our pipeline programs, but some of them may turn out to be bigger programs even than things that are in our pipeline. Now, with 18 things in our pipeline, we really don't need new programs, but there'll probably be a little bit of shuffling that goes on because these were programs we didn't think we could do a couple of years ago, and now we're making remarkable progress with them. So the CAR-T that we're developing, we've pretty much decided that really trying to make Prime an oncology company would not be a very strategic move on our part. And so it is a place where, for the use of CAR-Ts, where we can do some extraordinary editing, we probably will be mostly interested in partnering with others who really have the expertise in the space. On the other hand, CAR-Ts are also a very interesting approach, as I've mentioned once or twice before, for autoimmune or immunological diseases, where there's some great things that are just starting to be done. We think our CAR-Ts could have some advantages on what other companies are doing, but even those other companies are very, very early in the process, and it may be a place that we will think about, ourselves doing, and focusing on at Prime. I would point out that our general approach with BD for all of our programs is for the right partner. I don't want to sound like an advertisement, but we have too many programs and we don't have enough ability to do everything that we'd like to do. So for just about any program, we would have conversations, and we have had in many cases conversations with potential partners, because certainly before we're done, some of the programs in our pipeline, we will have to partner just because of, capability and bandwidth issues.
Got it. Thank you for also touching on the partnership aspect. I think that's also very helpful for us. Seems like we're out of time. I just want to thank you again, Keith, for walking us through the initiatives and progress with Prime Medicine.
Well, my pleasure. You asked great questions, and I thank the audience for their participation. So thank you, everybody. Bye-bye.